Welcome. I'm your host, Neil Howard, on Health Professional Radio. Glad that you could join us once again. In conversation this morning with returning guest, Dr. Doug Manion. When he was here before, we talked a bit about his work in HIV/AIDS therapies, a little bit of the personal connection. He's returning with us to talk about his cancer therapy work. Welcome back to the program, Doug. How are you?
I'm great. It's great to be back. Thank you.
Right. Dr. Manion, for our listeners who may not be familiar with you, when you were here before, give us a bit of background about yourself, and let's talk about what you're doing there at Kleo.
Thank you. So I am the CEO of Kleo Pharmaceuticals. So we're a synthetic chemical immune-oncology company. I'll explain what that means in a second. We're spun out of some excellent science done at the lab of Dr. David Spiegel at Yale, and we're based in the New Haven, Connecticut area. And what we do basically is with chemicals as opposed to biological drugs, we redirect the immune system to attack tumor cells. And my background, I'm a physician. I actually went into med school specifically because of the AIDS epidemic and wanting to have an impact on that disease, which I feel I did. I've 20 years experience plus in the pharmaceutical industry, working initially uniquely on HIV drug discovery and development, but in the latter stages of my career, I began to go into other therapeutic areas, including cancer. And I thought that there was a lot of similarities in terms of the impact on patients and my interest in cancer and HIV.
As far as cancer is concerned, are we talking specific cancers that Kleo Pharmaceuticals is working on, or all cancers? What specifically are we going to talk about this morning?
So our technology could actually be adapted to pretty much any cancer, which is one of the most exciting things about the approach we're taking. But of course, you have to start somewhere. And so where we have begun is on two different cancers. One of them is a cancer of the blood. It's actually of the bone marrow, called multiple myeloma. And the other one is one known to a lot of people, prostate cancer. But basically, the technology can be applied to either blood or solid tumors. The art is actually just finding the marker on the surface of tumor cells that our drugs can recognize and redirect the immune system against.
Are your compounds enhancing things that maybe other compound biologics that have been used with the patient? Are they enhancing? Are they replacing? How does it actually work?
Well, we actually have we have three different technologies. One of them would basically be to create something that works like it's a monoclonal antibody. So many companies create these antibodies outside of the body, so they're manufactured, and these are usually bioengineered to be a little bit different than the antibodies that we have naturally, and then they're given like drugs. So what we do for our first technology, called antibody redirecting molecules, is we have a chemical as opposed to a biological that's a bridge between the tumor cell and antibodies, which can basically redirect the antibodies people already have in their own bodies to act as if they were these ones that were created outside the body as a therapeutic. So basically it would be to provide the same effect, and I guess to compete against them. Now, there's a possibility in some instances that we could be complementary to those drugs because we can design our drugs to actually bind to a different area of the tumor cell than the antibodies do, in which case they could be used in combination. Another one of our technologies, called the monoclonal antibody therapy enhancers, or MATEs, actually is looking at partnering with companies that make monoclonal antibody therapeutics to actually enhance them. So with a chemical engineering process proprietary to Kleo, we can actually either improve their efficacy, or we can actually add on to them other means by which those antibodies can kill tumor cells.
Are both of these technologies primed for trials soon, or one or the other?
The first is, so we actually have two different arms, antibody recruiting molecules, that are currently going through what we call IND-enabling studies. So an IND is a pathway to the US FDA, an investigational new drug application. And so before you can test drugs in humans, you need to do a series of tests beforehand, and then you have to submit a dossier to the FDA. So we're actually preparing to do that for the first of our drugs, with the dossier being submitted this year. And so we'll be starting to treat patients as early as the first quarter of next year. And the intent would be to have the second of these drugs in the clinic no later than the end of the first half of next year. So we'll have two drugs in the clinic by next summer.
Prior to these trials, do you expect that some of these compounds will linger after they've run their course in the body? Will they redirect themselves, or will they just go dormant?
So we've designed our drugs very specifically to only have effect against tumor cells and not against any healthy cells. And so first and foremost, we want to make sure that we're only doing good and no harm. And we're doing a series of safety studies before dosing in humans to be as assured as possible. Of that, of course, we'll be monitoring safety in all of the patients who receive the drug once we begin clinical testing. The drug actually has a fairly long half-life, so we anticipate quite infrequent dosing every week, or less frequently yet than every week. But again, we'd be designed in such a way that we'd have no collateral damage. The only lingering effect would be a lingering effect against the tumor, which is exactly what you want.
Talk about some of the challenges that you overcame up to this point.
Yeah, so the drug hunting is a challenging pursuit for everyone, and so every drug before it ever gets to the market, probably like a cat goes through 9 lives where you think it's not going to make it, and so you have to be really clever to kind of work through all of the problems. So this drug actually has been relatively easy to date, although we anticipate that there could be bumps in the road as we move forward. You really do need to be resilient. I mean, the reason why they call it research as opposed to search is that you got to keep trying. You got to keep trying things until things work. So, for instance, the antibody recruiting molecule technology that we're now moving forward is a 3rd generation version of a platform that David Spiegel began to work on in his lab at Yale probably about 10 years ago. So although each generation had its own upside, there were still kinks to work out. And so the 3rd generation has solved what we think to be one of the biggest limitations in terms of this approach, which is to have enough antibodies to be able to redirect to have very clinically significant impact against tumors. And so with our new antibody binder, which we call a universal antibody binder, we've done exactly that. So we can now recruit the overwhelming majority of antibodies in the host with the tumor to be redirected against their tumor. And the good thing about redirecting endogenous antibodies, so antibodies that the person is making anyways, is that our body has a mechanism to replace them. So it's not like you're ever going to run out. So we can endlessly basically redirect antibody against the tumor and have a constant resupply by the host.
Give our listeners a website where we can go and get some more information about Kleo Pharmaceuticals and about your research there.
Yeah, please go to www.kleopharmaceuticals.com, and there you'll see a raft of information, including some publications and a really cool video that actually shows graphically how it is that our technologies work.
Right. And that's Kleo with a K, correct?
Kleo Pharmaceuticals.com.
Doug, thanks for coming back. Always a pleasure, and I'm hoping we'll talk again soon, man.
Thanks for having me. The pleasure is always mine.
You've been listening to Health Professional Radio. I'm your host, Neil Howard. Audio of this program is available at hpr.fm and healthprofessionalradio.com.au. You can also subscribe to the podcast on iTunes, listen in, download at SoundCloud, and be sure and subscribe to our YouTube channel.